Assalamu alaikum dear students. Today we are starting our new chapter, that is enzymes. It's your unit 3rd. So let me tell you first of all what an enzyme is. An enzyme is a biocatalyst that increases the rate of reaction without being changed itself in the overall process. Fine, an enzyme catalyzes a reaction but it does not change in structure in the overall process, and the molecules upon which the enzyme acts are called as substrates. Now let me tell you, here are some few properties of an enzyme which make them unique. Number one is most but not all enzymes are proteins. What it means, like all enzymes are made up of proteins, fine, globular proteins, but not all enzymes. Here is an exception, like ribozyme, that is not made up of protein, that is made up of RNA molecules, catalytic RNA molecules. So most of the enzymes you can say are proteins, but not all. Enzymes are highly specific, means enzymes are very specific in the reactions. They catalyze a particular type of the reaction and act on a particular kind of a substrate, which means they are highly specific in action and exhibit enormous catalytic power, means they have catalytic power. They can increase the rate of reaction. How they do it? They lower the activation energy. In this chapter you will be studying how they lower the activation energy. What is activation energy? Fine, so I'll be discussing these things later on in this chapter. So let me tell you, enzymes are of two types on the basis of the structure. Number one is simple enzymes and number second is conjugated enzymes. So what are simple enzymes? They are made up of only amino acids. They don't contain any other part. They are made up of only amino acids. And what are conjugated enzymes? These enzymes contain a non-protein group called cofactor. Fine. They have a protein part also and they have a non-protein group which is called as cofactor, fine, which is required for the biological activity. Like you must be remembering, in the splitting of the water in the photosynthesis, an enzyme is required and which contains which works in the presence of a cofactor, manganese. So what a cofactor does, it's required for the biological activity. Means a conjugated enzyme has a protein component plus it has a cofactor. Fine, it has a cofactor also, and this whole protein component plus cofactor, it's called as holoenzyme. It's called as holoenzyme. Fine. If the cofactor is removed, and this protein component alone, it will be called as apoenzyme. It will be called as apoenzyme. I hope this is clear. Protein component plus cofactor, this whole is called as holoenzyme. And if this protein component is removed, this cofactor is removed, this protein component alone is called as holoenzyme. So moving forward, let me tell you, एंजाइम्स जो होता है उसमें जो प्रोटीन कंपोनेंट होता है, अगर हम उसी को ले खाली, आई टोल्ड यू इट्स कॉल्ड एज एपोएंजाइम. इट इज़ जनरली बायोलॉजिकली इनएक्टिव. होता है एक्टिव भी कभी कभी, जनरली इट्स बायोलॉजिकली इनएक्टिव. फाइन. And let me tell you, this cofactors are simple metal ions. These can be simple metal ions like manganese is there, zinc is there, magnesium is there, and others are complex organic groups which are called as coenzymes. Means a cofactor can be simply a metallic ion. Fine. Metallic ion like zinc is there, magnesium is there, manganese is there, iron is there, and some cofactors are complex organic parts also, and those are called as, if they are complex organic groups, then this cofactor will be called as a coenzyme. I hope I am clear here. So cofactors which are tightly associated with the protein covalently or non-covalently are called as prosthetic groups. It is another thing you must remember. Those cofactors which are tightly associated with the protein component by means of a bond. That bond is either covalent or non-covalent. Non-covalent means ionic, it can be, fine. And so those cofactors are called as prosthetic groups. So let me tell you once again, the protein component alone is called apoenzymes, and generally these are biologically inactive. And cofactor can be a simple metal ion like zinc is there, magnesium is there, and Sometimes it can be complex organic groups, and if it is a complex organic group, then it's called a coenzyme. Fine, and I hope I'm clear here. And cofactors which are tightly bound either covalently or non-covalently are called as prosthetic groups. Fine, so moving ahead, naming and classification of enzymes. So what is there? Sometimes there's confusion, like let me tell you, ATPase. Fine, you have already heard this enzyme ATPase, ATP synthetase. ATPase is an enzyme which dissociates ATP, fine, which helps to dissociate the ATP into adenosine diphosphate plus inorganic phosphate. And ATP synthetase is an enzyme which helps to bind ADP and inorganic phosphate to form adenosine triphosphate. So there is a bit confusion. So what was does the International Union of Biochemistry and Molecular Biology have developed a nomenclature for enzymes. Fine, they gave the EC numbers. EC here stands for Enzyme Commission. EC stands for Enzyme Commission. For example, we have this enzyme hexokinase. Its naming was done as EC Enzyme Commission 2.7.1.2. So what is this 2.7.1.2? Let me tell you what is this. This first number, it is the class. It designates the class to which the enzyme belongs. It designates number second. It designates the subclass. And it designates number third, जो number है integer है, ये करता है sub class designate. It designates the sub class. And this fourth one, it designates what? Order. जिस order में ये enzyme इस category में add किया गया था. The order the enzyme was added in this category. So let me tell you, पहले हम enzymes के जो main 6 classes है, जो पहला है integer, वो class designate करता है. So we have 6 classes of enzymes. How hexokinase got its class? So we'll be studying. We have 6 classes of the enzymes. Number 1 is EC1, oxidoreductase. Oxidoreductase catalyze oxidation reduction reactions. These catalyze, what these enzymes do, these catalyze the oxidation and reduction reactions. And they include an extensive class of like dehydrogenases. They do what? They do hydride transfer. Hydride means H negative transfer. And oxidases are there, oxygenases are there, peroxidases are there. Here you can see A here, it's a reactant. Fine, it's reduced here, and B is oxidized here. And whenever the oxidoreductases act upon it, and they do what? They oxidize A and reduce B. Fine. So these are the class of the enzymes known as oxidoreductases. Now we have second class of enzymes. Those are transferases. What they do? They transfer a functional group. For example, a methyl group or a phosphate group. Here you can see, for example, the groups which they transfer are like acyl group, alkyl group, glycosyl group. Fine, between the two molecules. For example, here I have a reactant A and B. These are bonded together. Plus there is another reactant C. This reactant, this is a single reactant, and this is other. What they do, these transferases, they transfer a group. For example, they dissociate A and B, and what they do, they transfer this B group to the C. This is the reaction. So जो मैंने आपको starting में example दिखाई थी hexokinase की, hexokinase belongs to this class of enzymes. Here is EC2, means hexokinase is a transferase. Hexokinase is a transferase. And ये जो number second है, what it does, it designates the subclass. Like what type of reaction, ये क्या reaction किस bond पे attack करता है, it will show this. And number third shows the cofactor. Number third shows the cofactor, whether NAD is involved, when whether NAD positive is involved. It shows the third जो है इसमें, it shows the cofactor. And number last जो है, last, it shows the order, जिस ऑर्डर में ये इस कैटेगरी में ऐड किया गया था. मींस ये ट्रांसफरेसेस की कैटेगरी में सेकेंड एंजाइम है. Transferases की कैटेगरी में hexokinase is the second. So every enzyme has a proper nomenclature, so it will be like this. So for your understanding, I had chosen this example of the hexokinase. So number third category are hydrolases. What they do, they catalyze the hydrolysis of various bonds. What they do, they add water, fine, and help in the cleavage of the bonds. For example, these include esterases are there, glycosidases are there, lipases are there which act on the lipids, proteases are there. For example, A and B is a reactant plus water. What it does, A के साथ एक hydride ion and BOH, fine. So this is the type of the reaction hydrolysis catalyze. And number next we have lyases. Lyases cleave various bonds by means other than hydrolysis and oxidation. ना ये hydrolysis करते हैं, neither they do oxidation, and catalyze the breaking of which bonds? C to C bond, C to O bond, C to N bond, C to S bond. For example, इसमें category जो है aldolysis, which act on the aldehydes, aldolysis. Fine. So here I have A double bond B plus HX. Fine. What it does, it breaks this double bond, converts it into A bond X and B bond H. Fine. This is the type of the reaction, lyases cleave. And number next I have in this category are isomerases. What they do, they catalyze the isomerization reactions, fine, and includes epimerases, racemases, and intramolecular transferases. For example, here I have alpha D glucopyranose. This is alpha D glucopyranose, and it's changed into its isomer, that is alpha D fructofuranose. Alpha D, this is a pyran ring. Here you can see this is a pyran ring, and it has another isomer, that is the fructofuranose. It's changed by means of an isomerase into its isomer. So this type of the reaction is catalyzed by isomerases. And number 6th class is ligases. By ligases we mean joining. Ligases help to join. Like whenever you hear the word ligase, it means joining of the two molecules. And what it does, it uses energy in the form of ATP here, coupled with the hydrolysis of a nucleoside triphosphate. कोई भी nucleoside हो सकता है, it can be GTP, it can be ATP, कोई भी nucleoside triphosphate. For example, A and B I have here, plus ATP are used, and what it does, there is the joining of these. Ligase means joining. A and B are joined, and what is released? ADP plus inorganic phosphate, means it uses energy to join the different substrates together. Fine. So this is the third class of the enzymes, and I hope I am clear here. So if there are any questions, please let me know. Thank you so much. Stay blessed, stay safe.